Welcome class to Chapter 2, Biological Foundations, where I'm going to go over a little bit about heredity, prenatal development, and birth. Oh, skip the page. So what are the mechanisms of heredity? They are the chromosomes, and you have 23 pairs, which is 46. And basically, how did you get them? You got half from your mom and half from your dad, and so that means the human eggs contain 23 chromosomes, and the human sperm contains 23 chromosomes, and put it together gives you 46. Your whole body, all your cells, inside the middle, the nucleus of the cell, are 46 chromosomes. The only cells that do not have 46 chromosomes are your eggs or sperm, depending on which one you have, which only has 23. Here's the example right here. You got your autosomes, 1 through 22, and then your sex chromosome, and this is XY, which means it's a boy, woo! The zygote, that's what you were, and looks like your cells are already splitting right there and developing into that awesome student that you are today. Human egg and sperm unite, and it contains a complete set of 23 paired chromosomes, 46 in total. 46 chromosomes equals to be about 25,000 genes. And as I said, the autosomes and sex chromosomes, what they were, and if you're a sex chromosome, if you're a male, your sex chromosomes are X and Y. You got an X from your mom and a Y from your father, and if it's a girl, it's 2 X chromosomes, 1 X from mom and 1 X from dad. And then what are the mechanisms of heredity? DNA, or DNA if you are a Zootopia fan. DNA, each chromosome consists of one and only one DNA molecule. DNA is essentially information transfer, right? How does the structure know what to develop into? Information. So you're really information in a lot of ways. DNA contains the instructional code for cells to create amino acids, proteins, and enzymes, really the building blocks of your body. DNA molecule is a double-stranded helix, as you can see right here, of chemical compounds. The phosphates and the sugars make up the strands, and the nucleotide bases, which are made up of nitrogen, make up the stairs, and they are adenine, thymine, guanine, and cytosine. And the double-stranded helix consists of nucleotide pairings, so you'll always see the adenine with the thymine and the cytosine with the guanine. The order in which the nucleotide bases are structured is a code that causes the cell to create specific amino acids, proteins, and enzymes, like I said, which are the building blocks of who you are. So really the order in which these are structured is going to create your characteristics. Genes are any group of nucleotides creating instructions for a specific biochemical unit. Thus, they are the functional units of heredity because they determine the production of chemical substances that are ultimately the basis for all human characteristics and abilities. Let me just check, make sure we are filming now because I've been having some issues with the filming. And it is this time. Let's keep our double-stranded helix fingers crossed. Person's 46 chromosomes include a bunch of genes, 25,000 genes. I think I have it mixed up there. Chromosome 1 has the most genes, 3,000, and the Y chromosome the least genes at 200. Most of our genes are the same for all people, and fewer than 1% of genes cause differences between people. So we are all together, right? We're all one. We're all the same humans. Why can't we get along on this earth, you might ask? A couple terms. Genotype is the complete set of genes that make up a person's heredity, and phenotype is what you see, right? It's the individual's physical, behavioral, and psychological features. The interaction of genes on each pair of chromosomes produces traits. Each chromosome of a pair contains one parent's genes contribution to a specific trait. The alleles are different forms in which genes come. Each allele carries instructions for a specific variation on a trait. And in case of the shape of red blood cells, it's really a single gene inheritance. It's not a polygenic gene where there's multiple genes that cause this trait. There are 2 types of alleles that can be present on chromosome 11. One with instructions for normal red blood cells, and one to create sickle-shaped blood cells. Some more terms here: homozygous and heterozygous. Homozygous is parents give you a pair of alleles that are the same, and this typically results in the phenotype. And if instructions are for normal shape for both pairs, then you get normal shape for blood cells. And if it's the opposite, you'll get the sickle cell shape. Sickle cell shape. Say that 5 times fast. Sally sells seashells at the sickle cell shape blood cells or something like that. Heterozygous parents give you a pair of alleles that differ, and that creates a slightly more complex scenario in terms of what phenotype is going to develop. Thus, we have the terms dominant allele and recessive allele. Dominant allele means its chemical instructions are followed when both dominant alleles are present, and are followed when only one is present. So there's a dominant and a recessive. The dominant one's going to be followed. Recessive, though, means its chemical instructions are only followed if both alleles are recessive. And this kind of gives the diagram that simplifies it there. Unfortunately, for sickle cell disease, that is a recessive trait. And so the normal child will get the dominant trait, which is the normal red blood cell. And 2 of the dominant traits will produce a normal child, and then the heterozygous will predict what's something called, will create something called sickle-cell trait, and I'll talk about that in the next slide. And you have to have both recessive alleles to create the sickle-cell disease. And what I was mentioning was what's called incomplete dominance, a situation in one allele does not totally dominate the other. In a heterozygous condition, one dominant allele for normal-shaped blood cells and the one for sickle-shaped cells creates what's called sickle-cell trait. And this is a fascinating thing about how you can see variations in behavior or variations in phenotype. And you can see that here in this case where the gene is not active except in certain environmental conditions. So you can see how the environment and the genetic makeup can have some interplay there. Common phenotypes associated with single pairs of genes. I just thought that this was kind of fun and interesting to just look at dominant phenotypes and recessive phenotypes. You know, I have straight hair, normal hair, blonde hair, thick lips, or relatively, I guess. No dimples. Dimples? No dimples. No dimples is what it is. And so I just think it's kind of fun to just look at, you know, what phenotypes, what, you know, traits you have as related to the dominant or recessive alleles. 2 types of genetic disorders. There's the inherited disorders and abnormal chromosomes. Inherited disorders often involve 2 recessive alleles. And the examples are PKU, phenylketonuria, and sickle-cell disease. And then there's exceptions like Huntington disease, and that is on the that is a recessive allele. So one would wonder, how does it stay in the population and continue? And the reason is that there's an onset of the disease at a late time, like in the 30s. And so people end up procreating before they have issues related to Huntington's disease. The abnormal chromosomes, there's either extra, missing, or damaged chromosomes that result in abnormal development. The book talked about really mainly Down syndrome, also called trisomy 21, because there's one extra 21st chromosome. And they gave the example, if you give birth in your early 40s, you have a 1 in 50 chance of having a kid with Down syndrome. But it goes down significantly if you have a child in your late 20s. And then just some other examples involving the single X, XXY, or XXX, or XYY complement. Interestingly, no disorders consisting solely of the Y chromosome. The presence of the X chromosome appears to be necessary for life, which is kind of my theory is initially there were females, and then there was this random, you know, fluctuation in the genetic structure, some, you know, error that became males. The mutants are males, I guess. So the original Adam was Eve, I guess, something like that. So heredity, there's the idea of behavioral genetics, and these are researchers that study the inheritance of behavioral and psychological traits. Complex because most traits are a result of many genes, allele pairs, instead of only one gene or allele pair. And that's what's called polygenetic inheritance, when many genes affect the phenotype of a physical, psychological, or behavioral trait. Some terms right here. We've got dizygotic, or also known as fraternal twins. And those are two different eggs fertilized by two different sperms. So they are no more genetically similar than other siblings, except they're born in the womb, like giving each other high fives and stuff. But then there's the monozygotic, or identical twins, where you have the union of one egg and one sperm, but the zygote splits into two right after conception, and they are literally genetically identical. What do they like to do, those behavioral geneticists? They like to compare monozygotic to dizygotic twins to see how they differ on traits, and if they're more like the monozygotic twins are very similar on IQ or some other trait like height or something like that, then they might say, hey, there's a heavy genetic component to that, comparatively when they compare them to the dizygotic twins, who might not be as similar, for example. Adoption studies, you can compare children who are adopted to their, comparing them to their biological parents and then their adopted parents. A lot of people in the discussion talked about this and the fascination of how they see similarities in both, like their husband, for example, very similar to the biological parents as well as the adopted parents. And so also you can look at adopted twin studies and twins separated at birth. And so the book then talks about really this dynamic interplay, right? It's not nature versus nurture, but it's nature and nurture. There's this interaction approach with one really interacting with the other. And I think that's really fascinating how it's not just, clay is here, then the clay is shaped, but the clay is created initially, and then the environment can add clay on, remove clay, that type of stuff, which I think is really fascinating. I talk about taking the example of a seed. You plant it in good soil, provide it with sunlight, protection from diseases and other pests, and tend to it, water it, provide it nutrients, and it's going to thrive. Now, some plants do better than others, right? Some might grow bigger and stronger and stuff like that, and you can see the influence of genes. And genes can really set the limits on growth to a certain extent, right? But if you have this rad environment, it's going to help the best potential of that gene flourish, right? And if you have a pretty negative environment, even with those great genes, that individual plant might suffer in a lot of ways and even die. So you can see how that interaction is. Genes can sometimes, like I said, set limits on to a certain extent. Certainly the environment can cause genes to flare up or stay hidden, can create opportunities for growth or destruction. The best we can do for children is to provide them with the environment and experiences that allow them to thrive, and hopefully limit the negative effects if there's any dings on the genes, for example, of things that might flare up, but you provide them this awesome environment and then the genes just stay silent. So that's the idea there. Continued on this, we can control the environment and contain genetic expression, and think about PKU. If you diagnose that early and don't give the kid stuff with phenylalanine in it, then you will see no negative effects. Also, think about this concept. I really haven't talked about it too much, but the idea of neuroplasticity, that basically your thoughts, everything that, your emotions, all that are based on neural connections in the brain. And that neuroplasticity means that is plastic, that it can change, that learning through an idea of what's called growth mindset, where you challenge yourself to new problems and challenge yourself to work through an obstacle or something like that and learn something better. You're actually growing new neural connections. And so in some ways, you are changing what's thought of as a static thing, like intelligence, for example. Intelligence is not static. It can be, even though there is some genetic component to it, it's the idea that you can grow new neural connections and you're not limited by your genes. Epigenesis is the changes in phenotype not due to genes, and like I said, there's many influences of the environment, and like a lot of people said, how you're raised, and if you're raised in a nurturing environment versus a non-nurturing environment, how that can significantly change your brain development and the growth of a normal personality and not have a lot of psychopathology and stuff like that. But if you're in that negative environment, even if you have some super awesome genes, it may bring you down. The idea is the environment can trigger genetic expression. Think about how stress leads to menstruation at an earlier age, And the idea of methylation also, that neurochemistry changes due to environmental influences, that genes can be silenced or flared up depending on your experiences. But then there's also the influence of genes, right? There's things called the heritability coefficient. Intelligence, schizophrenia, for example, have an inheritability coefficient of 0.50. That means that in a group of people that those variations, those individual similarities can be explained by half of the essential relationship is related to genes, for example, the genetic influence of those traits. So why don't all monozygotic twins develop schizophrenia, for example? What factors of the environment might explain the differences? It really shows a dynamic interplay between genes and the environment, that even though twins, for example, they have a heritability coefficient, schizophrenia has a heritability coefficient of 0.50, obviously not all twins, monozygotic twins, identical twins, are both schizophrenic or the same in intelligence, that there's some other factors that might explain the difference, and that's one's environment. So genes can influence the kind of environment in which a person is exposed. I like to call it the double whammy. It's like, okay, you have, your parents give you these genes, and then your parents who have those genes, that gave you those genes, create an environment for you. So you get the double whammy. You get your parents' genetic influences, and you get the environment that they create through their genetic influences. There's also niche picking, and that's the idea that we seek out environments to confirm our identities, and so it ends up building kind of this self-fulfilling prophecy. The idea that smart kids will seek out smart kids, and that'll just kind of build on each other and stuff is the idea of that. So it's this dynamic interplay, right, between genes and the environment's on the phenotype. You can't actually change the genetic structure, but you can certainly change the way that your neural connections are happening or changing your behaviors and stuff like that, which can influence a lot of things that are thought to be static through what you've been given from your genes. Remember also that environmental influences typically make children within a family different. There are a lot of non-shared environmental influences as well, such as birth order. Well, that's kind of shared, but it's an environmental influence nonetheless. Personal experiences, psychosocial influences, and how we're treated differentially by our parents. So I'll probably just briefly go over this stuff. Don't want to make the video too long. It's like a pain in the butt to upload to YouTube. And you probably stop listening after about 10 minutes, right, is what they say. And so I'm probably at that limit. But be that as it may, this is kind of fascinating. The period of the zygote from conception to 2 weeks after fertilization, the zygote travels down the fallopian tubes and implanted in the uterine wall. Implantation triggers hormonal changes to prevent menstruation in the mom. In the center of the zygote contains the germ disc, and those are the cells that develop into the baby. So we were once a germ? What? That's a little weird. But anyway, other cells form placenta, where the nutrients and waste are exchanged between the mom and the child. And it's exchanged through the umbilical cord. Body structure period, the embryo is next, weeks 3 to 8. Here's some pictures of that. Body structures, internal organs, and 3 layers of the embryo develop. It's really starting to develop into what looks like a kid here, a little baby. And the ectoderm becomes the hair, outer layer skin, and the neural structures. The mesoderm becomes the muscles, bones, and circulatory system. And the endoderm becomes the digestion system and the lungs. Some more terms. It's inside this amnion, which is a sac filled with amniotic fluid, which contains the baby, keeps the baby safe, cushions it, and provides temperature control. The umbilical cord, as I said, connects the embryo to the placenta. And at this time, the neural tube swells to form the brain and spinal cord. And then there's the period of the fetus, and this is where the baby really starts to put on pounds, right? And you can see it in the mom. And just so many fascinating things are happening at week 9, differentiation between the ovaries and testes. And week 12, circulatory system begins to function, and you can start to feel movements in week 16. And other things in terms of viability is week 22 to 28, where the baby with a lot of help, obviously in the hospital, can live outside the womb. And then at week 22, the senses are active. Baby can remember voices, music. It's like rocking on to some Radiohead or something like that. It's like, I love Radiohead as a child. What is that? What's going on with that? Oh, maybe your mom listened to it. And then taste. Why do I like carrot juice? Because your mom drank carrot juice. And then just some important kind of things about nutrition, stress, and mother's age. Nutrition, it's really important that the mom get a lot of good nutrition, right? Eating the 5 major food groups and a lot of essential nutrients, iron, folic acid, to name a few. It's important to meet with a doctor to talk about necessary dietary supplements, proteins, vitamins, and minerals. You want to limit stress as much as possible, and better to have kids a little earlier than later, but not too early. Teenage girls are at greater risk to give birth early and to low-weight babies. The children tend to do worse in school and have behavioral problems. Success stories are there, but they're usually supported by the grandma. And older women have difficulty getting pregnant and less likely to have successful pregnancies and have increased risk for Down syndrome. Teratogens are agents that cause abnormal prenatal development, and there's a lot of bad ones out there. These are just some lists of some of them. In the area of drugs, you want to not imbue alcohol, not take aspirin, not take caffeine, and the other ones as well, of course. And they can cause big problems. Diseases can cause problems as well, and environmental hazards, making sure that you're breathing clean air. House does not contain products that have lead or mercury in them, not ingesting PCBs, experiencing x-rays, and then taking care of your cleaners, making sure that you don't have those bad cleaners, paint strippers, fertilizers, eating organic foods and non-processed foods. So keeping yourself healthy, because that baby is really sensitive to a lot of the things that the mom ingests and experiences. And then these are just some things about different impacts of the teratogens and how they can affect different structures and different aspects of prenatal development, and some information about genetic counseling and prenatal diagnosis, and different ways that you can get ideas about if your child is going to have any problems. Information about fetal medicine I thought was kind of fascinating. If there's some serious problems, what they end up trying to do with that. And the stages of labor, which are really just great and fascinating and fun. Stage 1, contractions become increasingly stronger and more rhythmic. Cervix enlarges to about 10 centimeters. Stage 2, the baby passes through the cervix to the vagina. Baby's head appears, that's called crowning, and the baby's born. And then stage 3, minutes after birth, the placenta is expelled. Just really important things to just highlight in terms of your chapter about the childbirth classes and how they can really prepare a family and a mom for the experience of childbirth. And adjustment to parenthood. It can be difficult the first couple weeks after birth. 50% of moms experience irritation, resentment, crying, reflecting stress and physiological changes, a lot of hormonal changes. And then months after birth, 10 to 15 experience PPD, which is postpartum depression. It may affect the baby negatively. Low self-worth, irritability, disturbed sleep, poor appetite, apathy, what the mom experiences. And risk factors are high hormonal levels, pre-pregnancy depression, stress, lack of support, and wonderfully that breastfeeding reduces that risk and gets that good connection, right, with the kid. Some birth complications are very serious. Hypoxia is one, and complications may result in doing a cesarean section or C-section, and then different weights indicating low birth weight, very low birth weight, and extremely low birth weight, where the poor little ones don't fare very well, and if they do, they have a lot of challenges afterwards. Then the infant mortality rate, just interesting how we could probably do better, a lot better, and it comes from having free prenatal care, inexpensive prenatal care, and paid leaves of absences for pregnant women. Hope this was helpful. Best of success on the quiz.